All right, so the field data that we're using for the last module here is a combination of in situ water samples, total suspended solids in milligrams per liter from water samples that we collected in October 2016. And corresponding with each of those samples is in situ water surface reflectance data from an ASD field spectrometer. So Alex went over earlier the water quality data that we collected from last year. So it's analogous to that data, but there's just a lot more of it now from last year's campaigns compared to the precursor campaigns. But same general process where we used bottle samples. We have 35 samples from this 2016 data set. And then this is the field spectrometer that we use to measure the surface reflectance, or the water leaving reflectance of each of those samples locations. And up in the right here, these are reflectance curves for different TSS concentrations. So you can see how much turbidity really impacts your reflectance signal, where there's much higher reflectance in the visible and up into the near infrared, the higher the sediment concentration is. So you can use that differing reflectance information to make a high quality model to estimate total suspended solids here.
Excuse me, I have a question for the slides. In the last slides. So yeah, for this figure, so this is a published work, or?
Yes, this is published work.
Okay, so for this color showed in this TSS in this figure, are they generated by the satellite or by the model?
Oh, by the model. Yep.
Like it's a reflectance model, right?
Yes, it's trained on the in situ reflectance data that was then scaled up to the AVIRIS-NG surface reflectance data.
Okay.
Yeah, from the pre-Delta-X, the 2016 field campaign and imagery.
So is there a particular reason it only covers this small region near the delta instead of, I know your original data is actually much larger than this small region.
So why are we just focused around the Wax Lake and Atchafalaya here?
Yes.
Oh, it's just the target area of those campaigns was much more focused around just the in the Atchafalaya Basin. There wasn't that much conducted around the Terrebonne for that campaign. The Delta-X campaign that was conducted last year 2 times over included a much broader area.
Also, it's not included in more open watered area?
Yeah, that's right. It was mostly just focused on the inland water bodies, the rivers and channels.
Okay, so I suppose the algorithm works for this region might not work for this more open water region. So is that the reason to only focus on this area?
I'm sorry, can you say that again?
So my question is, instead of including more open water region, more offshore the deltas, but only focus on this small region near the deltas, is that because these 2 regions cannot be analyzed together?
No, that's a good question. There's no reason they couldn't be analyzed together as long as you're accounting for the different water constituents that might confound your model a little bit. As you go out towards the Gulf, you might get more of a salinity gradient and different CDOM and chlorophyll concentrations that might skew your model results. But there's no reason why you couldn't study them together. It's just this campaign was really focused as sort of a test case over the hydrologic system in the Atchafalaya Basin, sort of a proof of concept for what we're doing with Delta-X right now. So we're mostly interested in the sort of variability in how much sediment is being output through those channels and how much of that might be being delivered to the wetlands.
Thank you very much.
All right, and lastly here, I just want to touch on how to apply these different regression methods for imaging spectroscopy data like AVIRIS-NG. So ordinary least squares regression, that's your typical regression method that you learn in Stats 101, right? But one of the assumptions that underlies OLS is that your predictor variables are independent, not collinear. And you also need a pretty large, robust sample size to make a good model. But that doesn't really fit hyperspectral data very well because, as we went over in the PCA module, those adjacent bands are highly collinear, so you can't use all of that information. But partial least squares regression is a method that works a lot better for hyperspectral data. You can use it to construct predictive models with many collinear factors because it doesn't rely on the same statistical assumptions that OLS does. And you can basically think of partial least squares regression as like a combination of PCA and OLS regression. It's like PCA in that you're deriving another synthetic set of components, but they're weighted towards towards the dependent variable that you're trying to estimate. So one other thing I wanted to note here is that another thing that having hyperspectral data with these adjacent bands lets you do is calculate the first derivative of reflectance. So that's basically just the slope of the reflectance curve. So how different one band is in magnitude from the next. So there's a couple different advantages to calculating derivatives. So first, it's less sensitive to intensity differences introduced by like BRDF effects and sun glint and things like that across your image. But it also makes any resulting model much more sensitive to absorption features like these absorption features in the visible ranges, and also the shape of these important slopes here. So there's this feature in the near infrared and then the red edge here that come out much more clearly when you calculate the derivative. So I'll go over this in more detail as we apply these data here. So we'll go back to the Python notebook. And the first thing we'll do here is open this CSV file that I have had uploaded into the Google Drive as well. So just to look over it real quick, this is what it looks like. There's 35 rows. The first column is TSS measurements from 2016. And then not sure what that was. But then there's resampled reflectance data for each of these AVIRIS-NG bands. So the in situ, the ASD field spectrometer, it measures reflectance in 1 nanometer chunks instead of 5 nanometers. So basically, we just resampled that to match the spectral response of each of the 5 nanometer AVIRIS-NG bands. So we have the estimated reflectances for each of those samples from 446 nanometers out to 1,000 nanometers here. So we'll load that one into our notebook here. And then this next cell is basically just a wrapper for the sklearn partial least squares regression function. So I have that one in a wrapper so that it just returns our predicted values, coefficients, the constant, the R-squared value, and then this other variable called a VIP, which is variable importance in the projection. So the next cell generates like a sort of first cut partial least squares regression model here. So we feed it in all the reflectance data here, sorry. And then the TSS. So this is the independent variables. And then this is the number of components. So I'm not doing any additional analysis here to like arrive at the optimal number of components. We're just feeding it the basic number, 2 components. So it's analogous to just like we did with PCA, making it give us 10 PCA components, except just 2 to keep it more simple. So the first thing it did here is then return the model R squared. So you can see it's a 0.81. So it's a pretty high quality model. And then the other thing I had it do is make this plot. So the blue line here is the average sample reflectance. And then the red line is the variable importance in the projection for each band that we input in the model. So it's sort of a self-explanatory parameter that we calculate concurrent with our PLSR model. And what it does is it basically tells us like how each band is weighted and incorporated into the model. So it's very useful for highlighting like which wavelengths specifically are most important for predicting TSS here. So for this model, you can see it basically highlights the whole visible, all the visible wavelengths, and then this near infrared feature, but not the whole near infrared range, just this bump here. So is that turning up for people if you're clicking along? So then this next chunk, given that the VIP scores can like identify what the most useful spectral information is, we just filter the bands down to reduce the number of inputs that we need. So VIPs over 1 are more useful, less than 1 we'll toss out. So it just increases the R squared a little bit, but it also reduces how many bands we actually need to like read in and take up memory later on when we actually apply the model and make our maps of TSS. So then this next cell, this is just to highlight the difference between reflectance and derivatives. So we use the NumPy gradient function on the reflectance data to calculate the derivatives of each sample. And so you can see, whereas if we're looking at these little absorption features in the visible, there's not that much of a difference in absolute magnitude between the trough and the crest here, right? But if you calculate the derivatives, it's much more pronounced. So calculating derivatives can really tune the model into those absorption features, and then these important transition areas where the shape might have a lot of influence on your dependent variable, right? So we trained that model based on derivatives as well here. And we see the R squared is about 0.816. And then the red curve shows the variable importance for that model as well. So you can see it's attuned to the, sorry about that. You can see it's attuned to those features, the red edge, and then this near infrared feature. The higher the spike, the more weighted it is in the model. So I wanted to introduce that concept as like another way of transforming hyperspectral data and another way you can get more useful information out of it. But that's another processing step in terms of actually having to handle your imagery and scale the model. So we're just going to use the basic reflectance model to actually like generate the map here. So the water mask path set here. So we're reusing that NDVI mask that we generated earlier in the second module, so that we're only taking the water pixels in. And then we're setting up an empty array. And then we'll add the constant from the model to those zeros as their sort of baseline. So this, I don't want to get too into the weeds here, but basically we need to like transform our image data into the remote sensing reflectance units that are favored by ocean color remote sensing people. So we apply a correction and divide by pi, just put in the right units, but again, I don't want to get too into the weeds on that. Just take it on faith that that's what you're supposed to do. And then you loop through each of these bands that we actually used in the model, apply that correction to transform it into remote sensing reflectance, and then multiply the band reflectance value by the proper model coefficient. And so we'll iterate that process through each of these bands. And then when we're done looping through everything, we have the final TSS estimate result. So that looks like a list form. And then if we reference the mask again, we can map those TSS values back into the water pixels. And it gives us our estimated TSS in milligrams per liter. So then if we save that, same way we did everything else, we can also bring that into QGIS. And then we have a map of sediment concentrations based on that model. So I want to note that approach is based on our published work from 2019. But the final model in that paper was based on the derivatives. And the advantage we found there was honing the model onto those narrow spectral features that I highlighted with the derivatives, made that model much more applicable to different study periods and areas so that we could actually transfer that model application across different sites and still return a well-validated result. The reflectance model here performed well too, but it was skewed a fair bit more compared to the derivative-based model. So basically, we're just like controlling a lot of those the water constituents that can skew your model by using the derivatives. But it's scaled up and derived exactly the same way as we just did here with the reflectance model. Question? I do not believe I uploaded that output, but I can. So this should be available as soon as this is done, if you want to take a look without having to run through everything. So I know I went through that one faster. I can tell everyone's getting rather exhausted at this point in the afternoon. But that's the last module, and I'll take any other questions before we wrap it up and take a break if anyone has any. Or if there's no questions, we can just go get more coffee.
Yeah, so we have a 15-minute break planned, but we are 20 minutes ahead of schedule. So we can still take a 15-minute break, and then Michael can start a little early.
I see one question in the chat from Glenn Swear. Are all these methods published by your group? So some of these methods are just established basic operations that we have applied them in our publications, but specifically for the TSS estimation, yeah, that algorithm was published by our group, yes. Question?
Oh man, I'm sorry, I forgot. Right into it. It looked like the TSS just broadly increases reflectance like down the board. Obviously, there was like a peak, like maybe in the IR or something like that. But
Yeah.
like you don't really. It seems like you don't really need the hyperspectral then. Like you could just really focus in on like one or two bands probably and really carry that same information.
Yeah, no, that's an interesting point. What you do still need is the correct narrow bands to be able to make those estimations. So for the final model here, this one more accurately reflects the final model that we actually applied in our paper. It probably only used, I want to say like 30 or so bands to actually capture these areas that designated as important. But you probably could do more analysis to hone that down even further. But what you couldn't do is just take multiple broad bands that cover like this whole broader region, right? But without actually capturing like this narrower absorption feature. So that's where the hyperspectral data comes in handy. We're actually deriving these optical properties.
Right. Actually, and that makes sense. And in fact, your final model was based on the derivative, which requires the hyperspectral. Yeah. Okay. Thank you.
Thank you. Another question back?
So is this similar, like when you do like a turbidity to TSS correlation, right? Like,
Yeah.
I guess you should, that relationship varies by location or time of year, right? Like it's very specific. Is this a similar thing where like, if you had, was this imagery, the AVIRIS-NG image taken at the same time that the field reflectance and TSS samples, or is this completely an independent
relationship? So there was we tried to overlap them as close as possible so that they don't all overlap within the ideal window of a couple hours of each other, but a healthy sample size did overlap so that we could verify like the measured reflectance from the field spectrometer paired pretty well with what the aircraft saw. So basically, yes.
My follow-up would be, so is this relationship that you developed from the field spectrometer, have you compared it to other, like if we would take satellite imagery from today, could you use this relationship, or is it like just?
So yeah, that's what we tested, and we found that we could actually. We took other data from the San Francisco Bay Delta Estuary and the Peace Athabasca Delta and just plopped the algorithm right on top of those data sets. One was another field spec data set, and one was from another similar sensor, hyperspectral sensor called PRISM. And they returned also very good correlations, well-validated results when we just directly transpose our model onto those data sets. But when we just transpose like the reflectance-based one, it's skewed a little more.
Okay, I have a question about this total suspended solid. I'm not very sure on the definition. I think while you are developing your model, you have the reflectance data and you also have field measurements for the TSS. And during the field measurement, does this include phytoplankton or just a sand or clay or just how would you define it?
No, it's all the solid material that's filtered out. So it's everything that comes within the, I can't remember what the how many micrometers the filter is good for, but it's so phytoplankton should still be captured within the TSS samples.
Does it make a difference if if like the sediment composition inside the solid are more sandy or more clay?
So we weren't able to get into that because our samples didn't disaggregate grain size that's definitely something that is a topic for investigation with the current data set. Yeah. But the 2016 data that this was based on, we didn't have that information. But it probably does have some bearing on it.
Okay, thank you.
A follow-up question, did you like divide the data like into inorganic and organic components of the TSS?
No, we didn't differentiate between that. I don't think the data itself had organic versus inorganic. We did have some other measurements, like chlorophyll A and chromophoric dissolved organic matter.
That's a different measurement, but from the TSS, did you differentiate?
Yeah, but we didn't differentiate between inorganic and organic in those data. It was just, yeah, TSS is like everything altogether.